Hi, Lindsay, you're on mute. Thank you.
Hello, sorry, I was on mute. Hello and welcome, everyone. Thank you for joining us today in this webinar, where we'll be sharing important updates on the latest edition of PS EN ISO 10993-1, requirements and general principles for the evaluation of biological safety within a risk management process. Our goal is to help you understand what's new and how your organization can align with these changes to ensure compliance and best practice. I'm Lindsay Ferrari, Lead Standard Development Manager at BSI. I have a portfolio of projects focused on medical devices, and I'm also the Committee Manager of the UK Committee on Biological Evaluation of Medical Devices. Next slide, please. Before we start, let me share some housekeeping. This is a listen-only webinar, as your mics and cameras will be disabled the whole time. Please submit any questions using the question button, and they will be addressed in the Q&A session. And if you have any technical issues, please report them through the same question button. The webinar is being recorded, and after completing the feedback survey, BSI will provide a link to the recording. This is a CPD accredited webinar. To request your certificate, please complete the survey as well. Next slide, please. Right, so I would like to introduce our speakers today. Michelle Kelly is chair of CH194, the UK Committee on Biological Evaluation of Medical Devices. She has worked as a consultant with NAMSA for 4 years, a leading global medical device contract research organization, providing medical device manufacturers with the support they need in navigating the complexities of medical device development. Prior to this role, Michelle worked as Biological Safety Manager for the MHRA. And in addition, she is a European registered toxicologist. Sophie Clulo works as Senior Biological Safety Assessor for the MHRA. She is responsible for providing expert advice and direction to the regulatory and safety and surveillance teams on compliance and vigilance matters related to biological safety issues. She is also Principal Lead Assessor on clinical investigation submissions for the specialist area of biological safety. She acts as MHRA representative for Committee CH194 and is also a European registered toxicologist and a member of British Toxicology Society. Welcome, Sophie and Michelle. I'm very, very glad to have you both here today. Sophie, over to you.
Thanks, Lindsay. All right. So to kick off this presentation, throughout this, Michelle and I will discuss the main changes to this document by framing the challenges in the previous version and how these have been or have begun to be addressed with the new version of the standard. So the first challenge is the checklist approach. It's fairly well known that Table A1 in Annex A of ISO 10993-1/2018 could generally facilitate a checklist approach to biological safety evaluations. Having that table standalone in the annex, as it did in the previous version of the document, kind of facilitated skipping through that essential written guidance straight to the tables. So this approach, as I've said, kind of misses that key information and guidance and sidesteps the risk management approach. So to kind of help align more closely with the risk management approach and ensure, reduce that possibility of missing key information and guidance, the categorization tables have now been integrated within the document. And you'll note there that I said tables, plural, and this is because the table that we all know so well has now been split into 4 separate tables according to contact type. So that's intact skin, intact mucosal membrane, breached or compromised surfaces or internal tissues other than circulating blood, and then circulating blood. So many of those contact types there, they'll be familiar to you from the subcategories of contact that were in the 2018 tables. And in general, these 4 tables still broadly look quite similar to the one that you'll be familiar with. But when you review the document, you'll see that there's a few different changes to it. One of them being the column that indicates the prerequisite requirement for physical and chemical information for hazard identification. That's now been moved out of the table and incorporated into the text, and that's because it's not obviously considered a biological effect. So that is another change you'll notice in those tables, the terminology change from endpoint to effect, and that's just because it's just more of an appropriate term. So there's some other changes that you'll notice to these tables that we go on to discuss a little bit later. Can you move to the next slide, please, Michelle? I think there's just a slight delay. There we go, thank you. Oh, it's just got to skip forward. Perfect, thank you. So another new section has been added to the 2025 document, and this is called Other Biological Effects. And these are effects that aren't listed in tables 1 to 4, but are effects that might be relevant to the use of the medical device. And these include immunotoxicity, neurotoxicity, repro and dev toxicity, and then material-mediated pyrogenicity. So the concept of these additional relevant effects isn't new. Annex A of the previous version did state that for particular medical devices, there's a possibility that that it will be appropriate to consider or include additional or fewer endpoints than indicated in that table. But this new section kind of brings this concept into the main body of the text and sort of promotes greater visibility of these potentially relevant effects. Consideration of these effects is generally either based on the materials used in the device and or the type of tissue contact that the device has with the patient. You'll also notice from this slide that MMP now appears as an other biological effect. And this is because MMP has been moved out of the table and obviously into the section. And this is because scientific literature has shown that chemically induced material-mediated pyrogenicity is rare and has been shown to occur only in a small number of substances, which aren't normally found as medical device constituents. And there is a reference to a paper that shows this in the new document. There's also another new section, and that's other factors to consider. And this includes medical devices with very brief contact, absorption through the skin or mucosal membrane, and then materials that change during use, including considerations of degradation, particulates, and toxicokinetics. So with regards to medical devices with very brief contact, this section provides guidance on like devices that typically have contact for less than 1 minute, and this could include things like lancets or hypodermic needles. And then it goes on to discuss the ability to prepare a written justification regarding negligible potential for biological harm. Then there's the section on absorption through skin and mucosal membrane, and this addresses situations where substantial absorption of constituents of a medical device is anticipated, and whether due to that absorption, if the potential consideration for systemic effects is required. And then we've got the other sections there that I've talked about, degradation, particulates, and toxicokinetics. They're not necessarily new, but this section, other factors, kind of brings everything together, all the information in one place that was previously kind of scattered throughout the document. If you can move on to the next slide, please, Michelle. Thank you. The next challenge is contact duration. So the description of contact duration categories was a little bit ambiguous in the previous version of the standard. The use of the term cumulative within the description has in some cases led to incorrect contact duration categorization. And that's been as a result of addition of seconds or minutes or hours of medical device contact with the patient. And this was never really the intention. So to help kind of remove some of this ambiguity, 2 new definitions have been added. So you've got total exposure period, which is defined as the number of contact days from the first to last use of the medical device, with a contact day being a day in which a medical device or component thereof comes into contact with tissues, including circulating blood. And that's irrespective of the length of time of that contact within the day. So if you've got contact within that day, it's a contact day. And this aligns with part 17 and removes the potential to kind of add up those minutes and those hours for the purpose of calculating contact duration. With that being said, though, obviously do keep in mind what I've just raised earlier about medical devices with very brief tissue contact. That concept can be applicable, so it's not the case that every single device that has some sort of contact in a day is at least a minimum of 1 contact day. So obviously you have got that very brief contact category as well. You could move to the next slide, please, Michelle. Thank you. Okay, so another new concept to consider when categorizing the device according to contact duration is the bioaccumulation of constituents used in the medical device. So this was not previously captured as part of contact duration calculation, and therefore long-term effects of bioaccumulative constituents that might have been left behind after a medical device use were not accounted for. So the new part 1 now states that the potential bioaccumulation of medical device constituents shall be considered for all duration categories, and states that where bioaccumulation is expected, the contact duration shall be long-term unless otherwise justified. So the key term here is expected, because at this stage in your categorization, you're not going to have analytical chemistry data. You're early on in your biological evaluation process. So this categorization is based on information you have available to you from the raw materials. That means from that information you've got on your raw materials, if you've got something that may be bioaccumulative, you're then considering additional relevant effects. So just bear in mind this change doesn't mandate extractable and leachable testing, but if you then go on to do extractable and leachable testing, because you need to obviously do so for a gap analysis purposes, and then you find something that might be bioaccumulative, you then might have to return back to that categorization stage, recategorize and consider if there are kind of any other relevant biological effects. You can move on to that next slide, please, Michelle. Thank you. So, sorry, just skip forward. Thank you. Another change is with regards to terminology. So you'll have probably noted, if you've had a chance to review, the term externally communicating has been removed from the tables. This term wasn't defined in the previous version of the standard, and what was meant by this wasn't completely clear, so it has led in the past to some miscategorization. And so to tackle this, the term has just been removed from the document, and the focus has now been put on the type of tissue contact for each component rather than just that category of device. And next slide, please, Michelle. Thank you. So the final change that I'll touch on is the implementation of a more consistent approach to genotoxicity evaluations. With the new 10093-1, genotoxicity evaluation is now applicable for all medical devices with prolonged contact, except for medical devices that are in contact with intact skin. In the previous version of the standard, genotoxicity evaluations were not indicated for devices in prolonged contact with mucosal membrane or breached or compromised surfaces, but were indicated for prolonged contact with other tissue types. So genotoxicity evaluations are now more consistent according to contact duration. The new standard, it does also clarify that the development of cancer by non-genotoxic mechanisms requires long-term exposure, and it's not expected to occur in prolonged contact. Therefore, for prolonged contact, evaluations for non-genotoxic carcinogenicity is considered unnecessary. So Annex B provides more information on the rationale behind this and other key changes in the biological effects listed in tables 1 to 4 if you are interested on reading up on this rationale. So I'll now pass over to Michelle.
Hello. So continuing on the changes or the challenges that we experienced with some of the terminology, as Sophia touched on, externally communicating medical devices, another terminology rather than anticipated or expected from the authors of the 2018 version. The word implantation effects was not very well understood, and actually I guess maybe we should have known that back then, because the word implantation definitely implies that it's only for those devices that are actually implanted in the body, but it was definitely expected that it's the local effects or that response that the device imparts on the tissue, and it's focused on the tissue contact, rather than necessarily just related to implantation. So it was interpreted incorrectly, and in the 2018 version we've included a phrase with regards to implantation effects, and while it says its implantation test can be used to assess local pathological effects, you can see that it is surgically implanted or placed in an implant site or tissue appropriate to the intended application. And actually they do state in brackets in the 2018 version an example of a dental application. So it was never meant to limit it to implants, but definitely the terminology wasn't helpful. So the 10.9, the latest version, was an opportunity to make this more clearer, and that's a simple change from implantation effects to local effects after tissue contact, has really made it clear that for some devices in contact with mucosal membrane, this terminology is even more important than certain devices that are implanted. So it's that inflammatory response or that local effects that your device has, that effect on the tissues, regardless of whether that's just skin, mucosal, or indeed implanted in the body, is now in scope. To aid in understanding this term further, there's now a new definition for tissue included in the definition section of the standard, of the new standard, including a note underneath the definition that really does convey the breadth of tissues that are under or in scope for this new requirement. So if a medical device has direct contact, and in terms of indirect contact, you have to think about any particulates that are generated as a result of use of that device. That is also considered a direct contact effect. Then you have to understand or assess the specific tissue or organ local tissue response that medical device has on the tissue, and that must be evaluated. So a little change like this in the terminology has now removed the ambiguity based on that was present in the 2018 version. As Sophia touched on a little bit, material-mediated pyrogenicity was, as we all know in this webinar, was quite a considerable endpoint. And for example, every single category from skin contact right down to, or surface contact, as we've often grouped them, down to implantation, all require this assessment for material-mediated pyrogenicity. And the working group tasked with changing the standard to what we have now, the 2025 version, really didn't feel that the science really backed up this, because while it's an endpoint that in certain circumstances absolutely needs to be considered, the science really was pointing to an effect that in some corners wanted completely removed from the standard, but perhaps the pragmatic approach right now was to limit it to a small number of devices rather than having it in the table for every single device. And it's often difficult to have the data to prove that it's not a requirement for your device. So invariably there was animal testing that perhaps wasn't required or relevant, and not sensible from an animal welfare point of view. And as Sophie had indicated, there's this new paper, Borton and Coleman. I would recommend the reference is in the new standard. I would recommend reading and digesting that information. And this paper naturally ask the question, is chemical induced material mediated pyrogenicity even an issue? And perhaps it's more a contamination issue or a manufacturing process issue rather than really a material mediated pyrogenicity issue. So in a way to combat that and really show that standards do change with time, while we know that we can't go to completely removing it completely from the document. It wasn't comfortable with the whole working group. We have a kind of a halfway house or a medium that sat with the working group was put together to say, let's not include it in the 4 tables, but it's now largely limited to novel materials, or of course materials that have or devices that have been shown to have a pyrogenic effect. I guess the novel materials is such that, do you have experience with this material such that you know what the pyrogenic response of that material is? So it's really trying to limit it to special cases rather than en masse that we do this material mediated animal testing test. So this was an opportunity to move with the science. And then, as Sophia touched on, this process is really trying to get away from the checkbox approach and trying to bring it into the risk management process. So there was a claim, and it was definitely expected or anticipated that biological safety would now sit within the risk management process. It was in the title, and a lot of that then the authors of the 2018 version of Part 1 expected that 1497 principles would be worked into it. A lot of the actual 14971 terminology was largely restricted to the annexes, however, and perhaps the main document only paid lip service to 14971. So while there was an expectation that some of the really important concepts within 14971 would come out to bear in a biological evaluation report, in practice that we needed to be more implicit or explicit about the overlap between the two. So there was a feeling largely that biological evaluation process was not really mapped across to the risk management process as laid out in 14971. So a huge amount of work was undertaken within the writing process for the new Part 1 in really mapping out the biological evaluation process and highlighting synergies or aspects where there was direct crossover, and perhaps other opportunities where there isn't huge crossover and an opportunity to make some of the risk management process more biological safety friendly. I'll touch on that in a little bit, but invariably that was always going to come with an increased number of concepts and terminology. So you will see the definition section within the new Part 1 has really expanded, and a lot of the risk management terms have now got the word biological before it, and it's fair to say within the working group consultation period when these documents were written, some thought that was a really good idea, and others perhaps, we should really be sticking absolutely to the risk management terminology and not altering them. And in a way, I think we came to a nice happy medium where some of the terms that we really needed, like biological evaluation, biological risk estimation, where we need the biological focus, so notes and guidance can be tailored accordingly, and then others, risk management, we haven't put the word biological, and all of that was over the years really thought of very carefully. And while there will be further iterations of this in the future, this significant change, I think does bring about this mapping between the two documents to really see. You'll see Figure 2 really does show you how the interplay between 14971 and 10093 Part 1 really does play out, and how the authors have really tried to make it as clear as possible. It's trying to see now, can we build up the guidance on how to use the terminology, but Rome wasn't built in a day, and I think the changes have been significant, and more will come in the form of guidance on how to use some of these terminology. So one of the aspects of 14971 that was the authors of the 2018 version, it was always meant to be inferred that reasonably foreseeable misuse, which is an absolute key input in the 1497 system, and personnel within a manufacturing organization that are so used to or au fait with this term for so many years. It was always inferred that this might come into 1093 part 1, if only because the title says it's within a risk management process. And it became obvious to the the people who were looking at the new part 1 and updating to the present version that we have, that actually it needs to be more explicit. So in 14971 it's absolutely called out. It's a very important concept. It requires manufacturers to consider any reasonably foreseeable misuse, and they put that down to predictable human behavior, which is a huge ask. And I don't know about you, but for me, trying to predict human behavior is quite a difficult task, and sometimes requires you to have a looking glass or predict the future. So it's not necessarily such a term that is easily understood. But the medical device regulations within Europe is also very explicit, and where you have intended use, you have reasonably foreseeable misuse. So whether we like it or not, it's a legal requirement. It's in the regulations. It's a requirement within 14971, which is one of the most important documents, overarching documents across your whole process, be it biological evaluation, clinical, regulatory. So it was incumbent on the authors of the new document to state whether should this be in or should it be out. And just like contact duration that Sophie had mentioned, where it was clear that the regulatory expectation was that you do not add up minutes or hours, when they were consulted on this part of the document, it became very clear to the working group within TC 194 that the regulators expected this to be considered. It was absolutely within the MDR, it's within 14971, and they saw no reason that it should be excluded. And for some regulators, they were surprised we were having the discussion. So this was an opportunity for the authors of part 1 not to make manufacturers' life more difficult, but actually to put some really real-world criteria on this term that actually would make it more user-friendly than the one that 14971 says, which is basically predict human behavior. So the definition is included. It is stated that biological safety is freedom from unacceptable biological risk in the context of both intended use and reasonably foreseeable misuse. But they have put some barriers or some criteria onto it. It has to be documented. So it's not that you just dream it up. So either, for example, if it's a legacy device, you will have some post-market surveillance showing that it's being used in this way. Or if it's not on the market currently, you'll have a very similar device being used in exactly the way that you would like your device to be used. And literature data shows, that's publicly available, shows that it's used in another way. And as well as being documented, it has to be systematic. So it cannot be just a rare one-off or a low number. There is going to be some guidance on what this looks like. But for now, systematic at least rules out those very rare cases. I don't know if that's going to be in terms of percentage. So if you've got a large number of devices and 1% could still be a huge number of devices, and a lot of this a discussion with your regulator on what they define to be systematic. During the development of this document, we were assured that this was a concept that was well known in the clinical world, and a lot of people use it in their clinical evaluation reports. But it was some terminology that the working group felt important to make the inclusion of reasonably foreseeable misuse much more user-friendly. So while I understand some misgivings about inclusion of this term, I think the trajectory or the way forward was always going to be understand what the regulatory expectations are for this term, now that we were going to make it more mapped or more aligned with 14971, and also a huge opportunity to make it much more user-friendly than the others. So perhaps a way of looking at this as a positive, even though I know it's a shock for certain manufacturers. Another one, well, lifetime was actually introduced in 2018, a requirement to evaluate your medical device across the whole life cycle. However, there was no guidance and there wasn't even a definition. So if you had a conversation about what they understood to be life cycle of a medical device, people would have severely or significantly different understandings. And if you don't have a starting point on what life cycle meant, it was very difficult to understand what guidance or what information you need to assess it. It's a huge topic, and there isn't a lot of guidance out there on this outside of the 10993 series. So this is a starting point. So the requirement is not new. So now we have a definition. So we know it's a severe series of all phases of the life for a medical device, right from when you initial conception to the final decommissioning and disposal. So this was developed through lots of consultations and worked on with the working group on what is understood by life cycle. So as well as the definition, also some basic guidance. I think it's fair to say that it's not significant guidance, and there are technical reports that are a report to be developed within the working group at speed to try and get this out and understand what it is. But at the moment, certain paragraphs within the document can give you an idea of what the regulators might expect when it comes to risk assessing a medical device for its whole life cycle. So the types of selection of how you select what materials you have, certain materials will lend itself to certain issues across the life cycle, and that might come into your consideration. Changes in the characteristics, example during transport, storage, or use. You might have more issues during transport and storage of a polymer, for example, compared to a metallic component. But it's not that the standard is requiring you to do all increased testing. It's just to consider as part of your material characterization exercise how your device might change, and not just focus on the device as it's coming off the conveyor belt. You have to do it right up to how the device is being used, and all of the changes that can happen during storage, packaging, and reprocessing might affect that patient who's going to be using, where the device is going to be used on. Reprocessing is probably one of the aspects that people are more familiar with, and we usually look at the maximum number of cycles a device can go through, the types of reprocessing agents, if they are toxic in themselves, or do they have a deleterious effect on the material. And of course, post-market surveillance data, this is not a new requirement, but there will be some instances in the literature, but aware of what types of things that can happen once your device is off the conveyor belt, is manufactured, but the post-market surveillance data is coming through to show that there's some effect, perhaps a physical failures or a breakdown or degradation of your material that's affecting the use of the material, or causing toxic issues. So all of these are sorts of things that get people thinking about the sorts of things that you might want to consider within your biological evaluation. But like I said, there is going to be more guidance to come on this, and I'm interested for one on what they come up with. But anything we were able to put forward for the new part 1 was going to be an improvement on just the bare sentence that is in the current 2018 version. So I guess one of the big things the 10993 series is such a huge series, is over 23 parts. What happens when they're forever changing? There is an opportunity for this standard to make recommendations on how regulators should almost like a transition period. What do you do with devices that are already on the market? And there is a paragraph within the new standard. And I think the most important message that the authors of this document wanted to get out was this should not mandate retesting. And I think it should not unnecessarily put burden on manufacturers to increase the testing of this device. And absolutely, there was an absolute hope that the vast majority of updates would be paper-based and really wouldn't generate the need for any testing. But I think as we've been with 10093 Part 17 now, that was such a big update. I guess the last biggest update compared to this one was that a gap assessment is probably the thing that you'll want to do with your devices, because what the guidance is asking you for is to ask yourself, or to determine, if all the changes that are in part 1, do they really increase the endpoints, or now, as Sophie said, we're calling them biological effects, or is it just around the edges, but actually we're changing the names of what they say, but actually the conclusion that we reached in our current biological evaluation report is absolutely still fundamentally the same, and there's nothing unsafe about that conclusion. And so you just review the historical data, and of course you're going to have much more data available to you than when you wrote the BER, because you will have all of that clinical experience if your device is on the market. So I think there's an expectation that hopefully the number of devices that are adversely or severely affected by part 1 shouldn't be any different, because I'm not sure this increases or what we were doing with the 2018 version. We're still the same people, we still knew what we were doing. A lot of people know how to do a biological evaluation, and we're doing it to best practice. This was almost formalizing some of the things that people had already shifted away from doing. I don't know how many people were really doing contact duration per minute and coming up with a limited contact device for chronic exposure. So I think there was a hope within the authors of the document and the working group that actually, you know, for the vast majority of devices, hopefully the amount of biological endpoints or effects won't change, and your biological evaluation report won't have to be updated accordingly. But the gap assessment needs to be somewhere documented, maybe an addendum to your biological evaluation report, but it's just demonstrating that you've done that job. If no additional biological effects, then an update is not required, but you need to show that you've done that gap assessment. If there are any changes, or you have to recategorize your device, then I think the expectation is you would use the data that you can gather, including clinical or any other data that you have to try and bridge the gap. If, for example, you have an endpoint that you really don't have data for example, genotoxicity testing, then unfortunately you might have to bridge that gap by generating data. And I think there's a hope that for whatever effect it is, that you would try and do in vitro or chemistry testing before you explore animal models, because I don't think anybody wanted this update to increase the burden on animal testing. If any changes and the last change that we've written here is no difference from the 2018 version. So if there's anything in your post-market experience that the device is now on the market and something's coming through that actually changes the biological evaluation, then you have to update your report. So that's nothing new. I think what the new is that your gap assessment to see if additional biological effects are going to be added. So by way of a conclusion, I think this is definitely, I don't know if we ever can get away from a simple checklist, but it's definitely moving further and further away from that risk of the old table A1 that we had in the 2018 version. The biggest changes, I guess, would be in categorization, the immediate changes. And for example, you might be looking at the way you add up duration now. You might have to consider bioaccumulation, or reasonably foreseeable misuse might be something that now is a requirement that you maybe didn't think about in the previous version, in the current version of your BER. And then, of course, what Sophia described about the biological effects or other factors. For example, do you now have the addition of genotoxicity for a prolonged contact with mucosal membrane, or do you have an additional biological effect that's listed? I think there are 11 or 12 of them that now necessitates the need for an additional biological effect. So those are probably the most acute changes. There will be others that come out, and technical reports should help us. But overall, I think these are probably the biggest changes that we have. So I'm going to hand over to Lindsay now, I think.
Thank you, Michelle and Sophie, for the presentation. Some interesting changes in the new edition, very insightful. Moving to our Q&A session. We've received a lot of questions. Thank you, everyone who submitted them. We will try and address questions as much as possible. So I'd like to start with the first question to you, Michelle. What is the direct impact of the new BSE and ISO 10993-1 to submission of old, current, and new devices?
Yeah, so I think to the old devices, I think the number one impact will be the requirement to do a gap analysis or a review of historical information to see if there's any changes. I think that's the biggest impact to the old devices, and I think preference should be given to those. Because depending on the size of your organization, you might have quite a lot of BERs to review. Probably preference to those BERs where there's a reasonable expectation that the number of biological effects will change, or there might be additional biological effects. So I think that's the urgent one for the legacy devices. For new devices, I think they're, while transition periods are being worked out, and that will be dependent on the regulator you're submitting to, there will be an expectation for new submissions that you will be working to the new Part 1. Of course, some of Part 1 we're waiting for further guidance, but in as much as possible that you will try and fulfill the requirements or the criteria within the new Part 1. I think for new devices, it might be less appealing for the regulator to accept working to the old Part 1, but for legacy devices, it will be your gap assessment. Absolutely.
Thank you, Michelle. The second one is for Sophie. What is the transition timeline?
Thanks, Lindsay. So a recommended transition period has been put forward of 2 years from the date of publication. And this is really because, as Michelle alluded to in the presentation, obviously, a lot of different guidance documents are falling out of this new revision. I think there's 6 or 7 different projects that have now come about as a result of this. So this 2 years is suggested to kind of provide enough time for users to kind of have that guidance to support implementation of the changes to the new standard.
Thank you. There is another one perhaps to you as well. Is there an estimation on the time point of FDA recognition, especially for the omission of testing material-mediated pyrogenicity?
As a UK regulator, I can't really comment on what the FDA timelines will be.
Yeah, I think this is probably much more uncertain than other standards. We know that it's a different political landscape within the US, and at the moment we're really not certain how it will be accepted at all. I think the FDA were involved in the process, and it's important to say that. I think for the vast majority of the stakeholders within Working Group 1, which was many different regulators, this is seen as an improvement from 2018 version. As Sophie said, we are waiting for technical reports to come in, really helping to understand how to use some of this new terminology. And I think us within the industry or within TC 194 absolutely expects the regulator or anticipates the regulator to be mindful, because they were so involved from the European and the FDA as well as the Japanese side on this document, that it does depend regulator to regulator, but I think there will be certain amount of pragmatism. But I think at the moment, what the only thing I can say on the FDA side is that their 2023 guidance remains a key document for submissions until we see the extent of recognition. But I think you'd need a crystal ball to understand when they might come out with their extent of recognition, if they come out in the short term at all. In terms of material-mediated pyrogenicity, it might be that you'll wait to see if your submission. I mean, I certainly am recommending rationalising some of this based on some of the science coming out and some of the guidance within Part 1. In terms of the appetite of whether the FDA will accept that, it might be that we have to understand based on submissions until the extent of recognition comes out. But I think it's really difficult at the moment for anyone to call whether they will come out with the extent of recognition in the short term.
Thank you, Michelle. The following question, let me read it for you, and we can see who can answer that. So, would an explicit risk evaluation be expected in the biological evaluation report now, as opposed to a reference to overall risk assessment for the device?
I'm not really sure what they mean by overall risk assessment for the device, but I think what the requirement within the Part 1, if you already have a biological evaluation report that's written and the device is on the market, is that there's an explicit documented gap assessment. So some recording somewhere that you've gone through your BER, you've matched it to the criteria within the current Part 1, and you've made a determination that there are no additional biological effects that need to be assessed, or no additional toxicological risks that have been identified. So how that's documented, we haven't stipulated within the new Part 1, but what is stipulated is that it needs to be documented somewhere so that it can be recalled as evidence. There is a requirement within the new Part 1 that if no additional biological effects, so you don't have identified any additional biological risks, then you don't have to go and update that BER necessarily. It's just this can be an addendum to your BER. Hopefully that answers your question, but no, there's no explicit requirement to go and redo your biological evaluation report just because there's a new Part 1, because that would be the case no matter what Part 10, 10.9.3 series has been updated, which is not really fit for purpose. So I think it's the review of the information and the documented evidence of that review is the really important key part here that 10.9.3 Part 1 2025 gets across.
Thank you, Michelle. We have many questions coming through. Next one, what is the impact to the risk management report based on ISO 14971-2019 with the publication of 10.9.9.3-1?
Yeah, this is quite an interesting question, and actually not one that's been posed up until now. I don't think it has any impact on 14971. Like I said in my presentation, I think the 10.9.3 Part 1 2025 has an opportunity to take some of the concepts, because this sits within 14971, but 14971 covers many aspects of your medical device. This is an opportunity for 10.9.3 Part 1 to make some of the concepts within 14971 more specific to biological evaluation. But I'm not sure this has any implications at all on 14971 and what's stipulated there. They should complement each other, and if there are differences, then it should be that obvious to the reader or the expert who's doing a biological evaluation report what the authors of part 1 were attempting to do, but I don't believe there's a feedback loop from part 1 that necessitates the change of anything to do with 14971. That's the parent document, and it was never any requirement or any understanding of the authors of part 1 that we would drive the process left. It was always 14971 driving or making us understand how we should write part 1 rather than the other way around at all.
Thanks. And regarding section 6.5.11.2, medical devices with very brief tissue contact, can you share more information on the justification and limitations to apply in this section? Are blood contacting devices also covered by this section?
So for this one, this is the one I touched on in my slides. When it comes to justification, it's around the justification for negligible biological harm, which will obviously vary device for device depending on how it's used, the materials used in it, the manufacturing process. So justifications are going to be based on that in context of that negligible biological harm. There are a few limitations that are written within that section. One is around the need to consider total exposure if you've got repeated use during a single 24-hour period. And then another limitation is around considerations of whether any materials are left behind after a medical device is removed. You then might need to consider a more detailed approach to your biological evaluation. And again, think about total exposure period.
Thank you, Sophie. Thanks for everyone submitting questions. Next one is, do you believe this publication will affect the calling of other known 10993 family biological risk standards, like, for example, the ISO 18562 family on biocompatibility evaluation of breathing gas pathways?
So always when one of the series gets updated, invariably it affects some of the other parts of the series. I'm not sure what the question referring to 18562. I'm going to give it a stab and say that perhaps the addition of total exposure period is now different because the 18562 calls on calendar days, so it's more risk averse than 1093 part 1, and that was huge amounts of discussion over 2.5-3 years by the leader of the author of part 1 on making 1093 or making the definition or how we calculate total exposure period to be a little bit more user friendly. I don't believe that anything in part 1 would drive a change in 18562. It doesn't make it more risk averse. It actually plays down some of the concepts, but it's up for, so every single document will have a convener, and when the document comes around to be changed, I don't believe part 1 will bring this process any more further forward, but when it comes up for renewal, as standards are constantly changing and evolving, then this aspect or this definition might change. That this is just, it's a very general question. It's just, I guess on my part, I guess of the significant difference between 18562 and 1093 part 1. But I know there's a move within TC 194 to have a definitions document and a drive in as best as we can we have equal or the same definitions across different standards, and perhaps their total exposure period will be looked at. But there's a different writing team and a different working group working on each document, and the consultation process will be different. So I don't believe it'll trigger it straight away, but for some documents they might consider some of the aspects within part 1 during their normal updating process.
Thanks, Michelle. I think we have time for a last one. Is there guidance on what competencies are required for the people performing gap analysis?
Maybe it's best I take this one. we did try within the working group to really understand what requirements do you need to be registered, do you need certain letters after your name, and it's really difficult to be that prescriptive. So I think the wording within part 1 is it's coming down to the education and the experience, and a lot of that will be the experience. So no, there isn't a hope or an expectation that prescriptive guidance from TC 194 will come out on this topic. What they do say is that you need to be experienced, knowledgeable, comfortable with the area that you're writing, and then there's a real expectation, given how complex the biological evaluation of medical devices is, that this will be a multidisciplinary process. So while I'm a registered toxicologist and I'm comfortable with certain aspects of biological evaluation, I'm certainly not going to put my hand up to, you know, going straight into a chemistry lab and starting to do analytical chemistry work. So it's based on what you're comfortable with and what part of the process you deem proficient. There is an expectation in your biological evaluation report that CVs will be as an addendum or the end of it, demonstrating an opportunity for the author of each biological evaluation report to demonstrate their competencies in doing that. But yes, there's an expectation, and it's called out in the current part 1 that this is a team effort. And it takes lots of different skill sets to bring a medical device safely to market for the benefit of patients. So it's your combination of your education and your experience in that area as to which part of that process you feel comfortable with. But we're not going to be prescriptive on what that looks like, because if you're too prescriptive, then you might knock out people that are far more experienced than people that have the letters after their name. And lots of people have examples of that.
Thank you, Michelle. Well, that brings us to the end of today's webinar. As I mentioned previously, you will receive a link to the webinar recording when it's available. BSE and ISO 10993-1 is due to publish by end of this month, but we might have a bit delay until first week of January due to Christmas season, but you will be notified when it's published. And we've got a special offer for you today. You can use the promo code ISO 10993 off and get a 15% discount at checkout on the BSI Knowledge once the standard is published. So I'd like to say thank you so much to our speakers, Michelle and Sophie, for the presentation and insights. I really appreciate it. And actually, thanks to all of you who have joined us today. Thank you for your participation and questions. And we hope you found the session helpful and insightful. Thank you very much and have a great rest of the day.
Thanks, all.
Thank you.